So the role of the medullary osmotic gradient is to facilitate that water reabsorption, not only in the loop of Henle, but it's also going to facilitate water reabsorption in the other distal parts of the tubule. So the distal convoluted tubule and the collecting duct. And this is going to depend on two things. One, the permeability of the epithelium to water, and then whether or not we've got water channels inserted in that membrane. Okay, so basically we cannot freely move water across our distal tubules. That's the distal convoluted tubule and the collecting duct. We cannot freely move water across that structure. This is happening dependent upon specific water channels, which we call aquaporins, that are inserted into the membrane of these parts of the tubule. So we have two aquaporins, aquaporin 3, which is always present in the basolateral membrane of this epithelial cell, and then aquaporin 2, which can be inserted depending on ADH. So ADH, or antidiuretic hormone, also called vasopressin. We talked about this when we looked at the cardiovascular system and our blood volume regulation. ADH is released from the posterior pituitary, and it's going to trigger the insertion of aquaporin 2 into the apical surface of the epithelial cells in these distal parts of the nephron. That is the distal convoluted tubule and the collecting duct, okay? And so when we have the membrane impermeable to water, water cannot leave the tubule. Again, it cannot freely move. It must have these channels, these aquaporin channels, in order to move through these parts of the tubule. And so more water will be excreted in the urine if we have no ADH in our blood, or if we have a membrane that is impermeable to water. Whereas in states of dehydration, in states of hemorrhage, in excessive thirst states, we have ADH that is flooding our system. Remember, it's a hormone coming from the posterior pituitary. It's going to go to the kidney, and it's going to stimulate the kidneys to insert these aquaporin 2 channels into the apical surface of these epithelial cells. And that is going to facilitate water reabsorption from the tubular structure, from the distal part of that tubule and collecting duct, back into the blood, okay? And so we can then regulate the concentration of our urine. And we can go from having urine that is about as dilute as 100 milliosmoles to having urine that is much more concentrated, which is about 1,400 milliosmoles. And so depending on our hydration status and other factors that I mentioned, we can regulate or manipulate the concentration of the urine, okay? Most of the water we absorb is dependent upon the length of the loop of Henle. And I kind of touched on this before, where we looked at animals that had really long loops of Henles, like our desert animals and camels, to other animals that have much shorter loops of Henle. And the longer the loop of Henle, the more opportunity for water reabsorption, and the bigger the gradient, right? The gradient between what's happening in our proximal convoluted tubule and what's happening deep in the deepest part of that loop of Henle. And so the longer the loop of Henle, the more opportunity for a bigger gradient and the more opportunity for more water reabsorption, or more water conservation, we can think of it as. Alrighty? Okay, so these two images kind of highlight that difference. So on the left, we can see what's happening if we're adequately hydrated, right? No ADH is in our system. We have no aquaporin channels in the membrane. And so the osmolarity of the tubular fluid stays the same because we're not reabsorbing any water. And as we can see, this osmolarity is going to be very low, meaning that the tubular fluid is going to be very watery, right? Not as concentrated, very dilute, and has a much higher volume. And you can think of this as being well hydrated, having lots of fluid in your system. And so you're not reabsorbing fluid because your body's not trying to hold on to water. And so you're really kind of flushing the kidneys out. You've got urine output that has a high volume and a low osmolarity. Now on the right, we can see an alternative scenario. This is going to be if you've got hemorrhage going on, if you're not drinking enough water in your diet, if you have excessive thirst or excessive sweating or a dehydrated state. And so we can see that the osmolarity of the tubular fluid drastically increases as we go from the distal tubule down through the collecting duct. So remember, this is showing the collecting duct here. And so as that osmolarity increases, we see that the urine output is going to be much lower in volume and much higher in osmolarity. And again, to make the connection here, higher osmolarity means we have a more concentrated urine. Think about a darker colored urine, more concentrated, less in terms of volume or quantity. And that is because we're reabsorbing most of the water as that fluid moves through the distal part of the duct, through the collecting duct here. And so this is why if you're not drinking enough fluid or if you're dehydrated, you have less urine and it's much more concentrated because your body's trying to suck all of that water out and hold onto that water because it is in a state of dehydration, okay? And this happens via ADH, which inserts those aquaporin 2 channels into the membrane. All right, I see a question here, so let me take a break. All right, so if someone wants me to jump back one slide, I believe it would be this slide here, or maybe this one. Okay. All right, so hopefully that was okay. All right, so let's keep going here. That is how we can regulate water reabsorption in the distal tubule and in the collecting duct. Now this brings us to speak about this obligatory water loss. Okay, this brings us to speak about this obligatory water loss that we kind of talked about a little bit earlier. So at a minimum, we must excrete 440 milliliters of fluid per day. That is what we call obligatory water loss. So even in our most dehydrated state, even if we're not drinking or bringing in any water at all, we still have to lose 440 milliliters of water. That is necessary to eliminate some of the waste material and some of these solutes that we have to eliminate, such as urea, such as creatinine, and some of the other solutes that we excrete. And so at that state, our urine is going to be at its maximum osmolarity. That's about 1,400 milliosmoles. And if you go back to this slide, we can see that in the instance where we absorb as much water as we possibly can, the urine will only get to an osmolarity of 1,400 milliosmoles, okay? So the window of the osmolarity of our urine goes from 100 milliosmol in its most dilute state to 1,400 milliosmol in its most concentrated state. Okay. All right. All right. So let's take a look. Actually, I'm going to skip over these checkpoint questions. We'll get back to these as we start next time. So take a minute to kind of review these as homework. And then when we meet on Monday, we'll review these. And if I skip over these, please be sure to remind me, just so that we don't lose the opportunity to go over those. Okay. So we're gonna look here at how ADH has an influence on that water reabsorption. So ADH is gonna regulate basically the permeability of the distal tubule and collecting duct, and that is going to manipulate the osmolarity of our urine, whether it's really dilute at 100 milliosmoles or whether it's really concentrated at 1,400 milliosmol. And this is this is brought about by those aquaporin 2 channels that are inserted into the membrane, okay? And that is under the influence of ADH, which we call antidiuretic hormone. And if you look at the name here, antidiuretic, right? if you understand what diuresis means, diuresis or a diuretic has to do with losing water. If you think about a diuretic, it's one of those medications that help us to lose a lot of fluid. And if we're going against that, anti-diuresis means that we're stopping water loss, or we're preventing water loss, or going against water loss. And that is the function of ADH, okay? It's gonna prevent or limit or inhibit the amount of water that we're losing through the urine. So as we wrap up here, let's go through this slide, which kind of explains how water is reabsorbed by inserting these aquaporin 2 channels. So first off, we start off with ADH as the stimulus for this water reabsorption. So again, think about states of hemorrhage, dehydration. We're gonna secrete more ADH into the blood. It's coming from the posterior pituitary, so there should be a review, right? ADH made in the hypothalamus, stored in the posterior pituitary, and released as a response to dehydration or hemorrhage. And so it's going to flood the blood, it's going to go down to the kidneys, and it's going to bind to one of these receptors that is on the epithelial cell of the distal convoluted tubule and the collecting duct. When it binds here, because it's a metabotropic receptor, we know that it's going to be coupled to a G protein. That is going to facilitate the production of some second messenger. In this instance, that's going to be cyclic AMP, and that is going to cause the activation of protein kinase. And protein kinase is going to synthesize these aquaporin 2 channels that are required in order to reabsorb water. Okay, and just to remind ourselves of the orientation here, this is the lumen of our tubule. So this is where we've got the water that's sitting in our tubule, the filtrate, rather, that becomes urine. And then on this side, we've got our plasma where we want to move that water to. Now, we talked about 2 different aquaporins. One is aquaporin 3 that is always present on the basolateral membrane. And then aquaporin 2 is not always present. It's only going to be synthesized in response to ADH release. So ADH synthesizes the second messenger pathway that creates these aquaporin 2 channels. They're going to be transported to the membrane, inserted into the membrane, and that is how we can facilitate water movement from our tubular lumen into the cell. And then because we've always got these aquaporin 3 channels on the basolateral membrane, water can simply move through these channels and into our plasma. So this is how ADH regulates the reabsorption of water in the distal tubule and in the collecting duct by inserting these aquaporin 2 channels into the apical surface of these epithelial cells. Okay. And so that is how ADH regulates water reabsorption in the distal parts of the tubule. And so this slide kind of summarizes that, the fact that ADH secreted from the posterior pituitary, right? These are cells that are originating up in the hypothalamus. Remember, the posterior pituitary doesn't actually make hormones itself. It simply stores those hormones that are made in the hypothalamus. And that neural connection where we have those axons making the hormone in the hypothalamus, storing the hormone in the axon terminals of those neurons, and then releasing it on response to certain stimuli. So we have certain centers that are in our hypothalamus. These are called the osmoreceptors receptors in the organum vasculosum of laminae terminalis, a very complicated and strange name, which we sometimes refer to as OVLT. These are basically receptors that are sensing the osmolarity of our plasma. And they're basically going to determine whether or not we need to secrete ADH. They're going to be in areas where we do not have the blood-brain barrier. So if you remember the blood-brain barrier, which is another important structure we talked about in a previous unit, the blood-brain barrier kind of sits between what's happening in our bloodstream, in our plasma, and what's happening in our nervous tissue within the brain. And so it's going to kind of interrupt or block an accurate sense of our plasma osmolarity. And so we don't have OVLT receptors in areas where we have the blood-brain barrier. The areas where we have these receptors is not interrupted or blocked by having the blood-brain barrier present. And so therefore we can get an accurate measurement or an accurate representation of what our plasma looks like in order to adequately secrete ADH or not secrete ADH in response to the osmolarity of that plasma. And so ADH is also affected by our baroreceptors. We talked about baroreceptors, basically those stretch receptors that are detecting our blood volume and blood pressure. And so if we've got stretching happening on those baroreceptors, that is an indicator of increased blood volume, increased blood pressure, and so we decrease ADH secretion. But if we have decreased baroreceptor activity, meaning not an adequate amount of stretch on those receptors, then we want to increase ADH secretion because that is a signal of decreased blood volume. That is a signal of inadequate perfusion, inadequate blood pressure. And so we need to reabsorb water in order to maintain our blood volume, and we do so by increasing our ADH secretion. And that ADH secretion, we talked about going to the kidneys, inserting those aquaporin 2, and bringing back water into the plasma. And then this flow chart kind of summarizes that phenomenon as well. So increased osmolarity of our extracellular fluid sends a signal to the hypothalamus, which detects that increase in osmolarity. And I keep saying osmolarity, but I want you to be thinking about that in terms of being more concentrated, being more salty, and being less dilute. And as we have an increased osmolarity, those receptors detect that. They signal the hypothalamus to make more ADH. That signals the posterior pituitary to release that ADH. The kidneys will get that ADH, and the kidneys will basically, the epithelial cells of the distal convoluted tubule and the collecting duct will insert those aquaporin 2 channels, facilitating more water reabsorption or less water excretion, and that is how we can conserve body water in the face of decreasing blood pressure or decreasing blood volume. And then that will obviously turn off this initial signal as a negative feedback response, okay? All right, and then this other flow chart here is showing how the baroreceptors are also playing a part in this. So drop in mean arterial pressure, drop in blood volume, which are the stimuli of hemorrhage or dehydration, will decrease our baroreceptor activity. That will also send a signal to the hypothalamus to increase ADH production, which will increase the secretion or release of that ADH from the posterior pituitary, and that will have the same effect on our kidneys. Increase water reabsorption, decrease water excretion so that we're not losing excess amounts of fluid, and conserving of our blood volume, and then that will turn off that initial stimulus of decreased blood volume and decreased mean arterial pressure.